Hello students, this is Geethanjali. In this video, we'll be learning about Mendelian disorders. So, in general, genetic disorders are categorized into Mendelian disorders and chromosomal disorders. In Mendelian disorders, we find defective genes due to which abnormal characters are seen, whereas in chromosomal disorders, the number of chromosomes are abnormal. So, let us see a few Mendelian disorders. First one is color blindness. So, color blindness is an X-linked recessive disorder. That means the defective gene responsible for this disease is present in the X chromosome. So, in this disease, the person cannot distinguish between the color red and green because of its defective cones. Okay, so we know cones are responsible for color vision. So, they have defective cones and they cannot distinguish between red and green, and you can see it occurs in about 8% males and 0.4% females. So you can see males are more prone to this disease than females. So why is that? Since we know this is an X-linked disease, females have two X chromosomes. So for the disease to express itself, both of them should be defective. Okay, whereas in males, there is only one X chromosome. So even if one X chromosome is defective, it will express its trait. So it will be defective and the male will be affected. So that is why it is more common in males than females. Coming to another disease that is also an X-linked recessive disease, that is hemophilia. So hemophilia is called the royal disease because it runs in the family of Queen Victoria of England. Okay, and in this disease, faulty blood clotting is seen. Okay, so whenever a person gets cut, the bleeding is continuous, okay, because the blood clotting is affected there. So that is why it is called the bleeder's disease, as continuous bleeding is observed. Coming to another example, sickle cell anemia. So we saw sickle cell anemia in one of the examples of frame shift mutations. So this is also an autosomal recessive disorder, and it is controlled by a pair of allele. So as I told you, it is autosomal recessive. That is why the heterozygous condition and the homozygous condition of the normal gene both lead to a normal condition, whereas the presence of two defective alleles will lead to a different condition. So let us see what actually happens here. So in sickle cell anemia, the sixth codon, okay, of the beta globin chain has a mutation. Okay, so what actually happens is that in our original gene, which is present in the nucleus, there is a substitution and the adenine is replaced by thymine. Okay, and that is why the mRNA that is transcribed from this gene is also different. Okay, so the codon here is GAG in the actual, but instead of that, GUG is present. Okay, so in that case, when the peptide is formed, okay, during translation of the mRNA, instead of glutamic acid, valine is produced. Okay, so because of this production of valine in this peptide chain, in this beta globin chain, the cells kind of look like a sickle, and that is why it is called sickle cell anemia. The red blood cells, their shape is affected. Okay, and that is why we call this a qualitative problem. It is not a quantitative problem. So qualitative means the amino acids that make up this chain is faulty. Okay, they are that is the valine is not supposed to be there. Instead of that, glutamic acid is supposed to be there. That is why it's a qualitative problem. Now, moving on to the next condition, phenylketonuria. So we saw this as an example of pleiotropy, and this is also an autosomal recessive disorder. So in this condition, The enzyme phenylalanine hydroxylase, which is responsible for the conversion of phenylalanine to tyrosine, is absent because of the defective gene. So, because of this, the phenylalanine is not converted to tyrosine and it gets accumulated, and this accumulation leads to the formation of phenylpyruvic acid. So, this phenylpyruvic acid has multiple abnormal undesirable effects. So it causes mental retardation, okay, hyperpigmentation, and also the kidneys perform very poorly, which causes minimal absorption and etc. So this was in short about phenylketonuria. Next is thalassemia. So in thalassemia, there are two types, okay, alpha thalassemia and beta thalassemia. So we know that hemoglobin consists of two chains, two alpha chains and two beta chains. So when the production of alpha chain is affected, we call it alpha thalassemia, and when the production of beta chain is affected, we call it beta thalassemia. So the alpha peptide chain production is controlled by two pair of genes. Okay, so these two pair of genes are HBA1 and HBA2, which are present on the chromosome number, which are present on the chromosome 16. Okay, so as there are two genes responsible for this, that is why the mutation can happen in any of the four alleles, or all of the alleles, or more than any four alleles, or there can be more than one mutation also. Okay, so but in beta thalassemia, the beta peptide chain production is controlled by only one pair of genes, which is the HBB, and it is present on the chromosome 11. Okay, and since it is only one gene, there are only two alleles, so the mutation can happen in either one of the alleles or both of the alleles. So thalassemia, unlike sickle cell anemia, is a quantitative problem. Okay, in sickle cell anemia, the amino acid constituting it was wrong. Okay, whereas in this case, the amount of production, okay, the amount of beta peptide chain production and the amount of alpha peptide chain production is affected. So in this case, we call this a quantitative problem and not a qualitative problem. So coming to an example of autosomal dominant disease, that is myotonic dystrophy. So in this disease, progressive degeneration of the muscles is seen due to prolonged muscle spasms, and a lot of muscle weakness is also seen. So this is the only example of autosomal dominant, that we should remember. So these were a few Mendelian disorders.